Hi everyone, and welcome to this presentation entitled Inferring Cell Type Mechanoregulation During Bone Fracture Healing Using Spatial Transcriptomics. We are interested in bone adaptation and regeneration, and we have been focusing on mouse models which allow us to investigate how macroscopic events like mechanical loading act on bones. And we aim to understand how this is transferred across scales and influence osteoblasts and osteoclasts to form or resorb bone accordingly, completing what is known as bone mechanoregulation. Experimentally, different methods can be used to analyze each scale of this complex tissue. At the tissue level, we have microcomputed tomography imaging, which can be performed in a time-lapsed fashion to identify bone remodeling events. And with microfinite element analysis, we can recreate the in vivo mechanical environment, allowing us to associate remodeling events with mechanical signals. This is the basis of tissue mechanoregulation analysis. At the molecular level, recent spatial transcriptomics technologies can now provide unbiased gene expression maps across the tissue while preserving the spatial context in 2D. So we aim to profile the local in vivo mechanical environment of bone cells, which we collectively describe as spatial mechanomics. And we can achieve this through our recently developed multimodal imaging approach called spatial microprobe. For this study, we consider a mouse femur defect model, where mice are loaded and scanned weekly with micro-CT, and focus on a control, sham loading scenario and a loaded condition from which we obtained an ST section at week 5 post-surgery. So we can infer relative cell type proportions for each location in the section, which we can model with generalized additive models, producing cell type-specific mechanoregulation curves. Under loading, the proportion of osteoblasts consistently increases over time, with a stable participation of osteoclasts in the same period. Spatial microprobe allows integrating 2D and 3D imaging data to quantify the live profiles of cells involved during bone adaptation and regeneration, enabling a new understanding of mechanomolecular responses in bone biology.